Hello, my name is Avery Fulford, and I recently graduated with a Bachelor of Science from Duke University. I'm currently working at NASA with Dr. Heather Graham before I apply to PhD programs in microbiology this fall. Today, I'll be discussing my senior thesis, entitled A Comparative Genomic Analysis of the 250 million-year-old halophilic archaeon Halosimplex carlsbadense with related species. My mentor for this project is Dr. Amy Schmid. So, in the year 2000, microbiologist Vreeland et al. Extracted salt crystals from a halite formation deep underground in New Mexico. These salt crystals contained pockets of fluid which are believed to be the rare liquid remnants of a Permian ocean which evaporated 250 million years ago. As seen in Figure 1, these halite crystals are pictured, and also the fluid inclusions are marked with the letter I. Using extensive methods to prevent sample contamination, Vreeland et al. Swabbed petri dishes with these brines and found that 4 microorganisms which were living within the brines were able to grow and form colonies. Because the microbes lived in brines enclosed in salt crystals for these 250 million years, they are quite possibly the longest living organisms studied to date. While there has been some controversy surrounding one of the bacterial samples, called sample 293, another microbe of the 4 extracted has been widely accepted as a new species and genus named Halosimplex carlsbadense. This organism is a halophilic archaeon. So to define these terms, a halophile is an organism adapted to highly saline conditions, and an archaeon is a microorganism in the third domain of life, the archaea, as opposed to the bacteria or the eukaryota. The overall goal of this study was to identify the physiological adaptations that enabled H. carlsbadense to survive in the harsh environment of a salt crystal over 250 million years. We achieved this by aligning its genome to the genomes of 17 other halophiles to identify homologous genes, defined as genes inherited by 2 species from their common ancestor. Homologous genes that are highly similar between 2 different species are said to be highly conserved, which suggests that they encode an important function for the cell. To move on to a description of our methodology, we use several computational tools to align and compare the genome of H. carlsbadense to the genomes of 3 groups of organisms. The first comparison group included 2 recently discovered species within the Halosimplex genus, H. rubrum and H. pelagicum. The second comparison group was composed of the 13 most closely related species of archaea to H. carlsbadense, which were identified in a phylogenetic tree created by Becker et al. In 2014. This tree is replicated at left in Figure 2, in which you can see H. carlsbadense highlighted in yellow. Each node, or fork in the branches, represents the last common ancestor shared by 2 groups of species before they diverged. Confidence values for each node are written below. These are called bootstrap values, which indicate how many times out of 100 repetitions the same node was recorded when repeating the phylogenetic construction on a resampled set of the same data. The third comparison group we chose included sample 293, a bacterium which is another of the 4 samples extracted by Vreeland et al. In 2000. We assembled its genome from short nucleotide sequences using SPADES software. Okay, for my results section, I'm first going to talk about the genomic alignment and comparison of H. carlsbadense and the 2 other Halosimplex species. In this alignment, we found that there was one long homologous sequence which was highly conserved among all 3 Halosimplex species. This was much longer than the other homologous sequences identified. This long homologous region encodes 70 labeled genes of known function, as well as 93 hypothetical genes whose function has not been categorized yet. We grouped the 70 labeled genes into categories based on gene function and compared the number of genes in each functional group in the longest homologous region between Halosimplex species to the number of genes in each functional group in the entire H. carlsbadense genome. This can be seen in Figure 3. In other words, the green bars represent the percentage of all genes in the H. carlsbadense genome which are involved in stress response, nucleotides, so nucleotide synthesis, DNA repair, and sometimes DNA replication, etc., and membrane transport. The blue bars represent the percentage of all genes in the longest homologous region among Halosimplex species, which are involved in the same three categories. While there are many more variables we studied, I've included the three with the greatest difference between the percentage of genes involved in each functional category in the entire Halosimplex genome and the longest homologous region. To summarize, Figure 2 suggests that the longest homologous region between the Halosimplex species is enriched in stress response, nucleotides, and membrane transport mechanisms, which could mean that these functions are important for survival in hypersaline conditions. This is because these organisms are highly adapted to high salt brines. Now I'm going to address the second genomic alignment and comparison we performed between H. carlsbadense and its 13 most closely related species of halophilic archaea. This alignment pinpointed 32 homologous genes within 20 clusters, which encode highly conserved proteins involved in oxidative stress response, DNA replication, and respiration. Oxidative stress is caused by hypersaline conditions. It's defined as a disturbance in the balance between the production of reactive oxygen radicals and the cell's antioxidant defenses. Now, for the sake of time, I'm not going to elaborate on all 32 genes involved in this alignment. However, two interesting genes are LonB and SUFB because of their function, which is highly specialized in halophilic archaea. LonB removes misfolded proteins from cells, which are damaged in hypersaline conditions due to oxidative stress. SUFB repairs cellular iron-sulfur clusters, which are also damaged by the oxidative stress of highly saline brines. These iron-sulfur clusters are incredibly important for the cell because they are required for the function of proteins involved in a wide range of activities, such as electron transport in respiratory chain complexes, regulatory sensing, and, most importantly for this analysis, DNA repair. Finally, the third genomic comparison we performed was between H. carlsbadense and bacterial sample 293, which, again, was another of the 4 ancient samples extracted from salt crystals by Vreeland et al. In 2000. Surprisingly, we found a 2,545 base pair long homologous region between the genomes, as well as 7 isolated homologous genes. This is a very high number of homologous regions for 2 species from separate domains of life, the archaea and the bacteria. This is suggestive of horizontal gene transfer, which is the transfer of DNA from one organism to another without inheritance from a shared parent. It has been demonstrated before in the literature that hypersaline environments make some species more likely to accept DNA from other organisms, even if that organism is from another domain of life. This is because of the broad-scale competency exhibited by stressed cells. The homologous genes between H. carlsbadense and sample 293 are represented, though not to scale, in Figure 4. The blue lines in this figure represent the genomes of the 2 species, and the colored boxes indicate homologous regions which are connected between the genomes by a line of the same color. Some homologous genes of interest are the 5 DNA repair proteins in one cluster, as shown in the teal box. There's also the ABC transporter, GLTS, and DUF domain protein, which all encode intermembrane transporters. There's also ACE-F and PEP-F genes, which are both metabolic enzymes. In conclusion, gene conservation across this Halosimplex clade, closely related halophilic archaea, and the halophilic sample 293 suggests that DNA repair, membrane transport, and oxidative stress response mechanisms are required for the survival of organisms entrapped in hypersaline conditions over long periods of time. DNA repair, for instance, can combat the damage that hypersaline conditions wreak on DNA, particularly through the rearrangement of bonds in adjacent pyrimidines. Membrane transport proteins, such as potassium and sodium transporters, protect against salty, hypertonic conditions, which can cause a cell to shrivel up and die through desiccation. Finally, oxidative stress response mechanisms prevent protein and membrane damage. We've identified 3 important avenues for future research on this topic. The first is the identification of genomic sequences in H. carlsbadense that did not align to related species, because genes that are unique to this ancient organism could encode novel physiological traits. Future research could also study 2 hypothetical proteins that we identified in our alignment of the 14 closely related species of halophilic archaea. It is rare for a hypothetical protein to be so highly conserved amongst a large group, which suggests that these proteins could encode an important physiological function that is not yet characterized. Finally, future researchers could investigate the possibility of horizontal gene transfer between H. carlsbadense and sample 293 using 2 methods of species-specific genomic metrics. These are species-specific codon use and GC content. To conclude, I'd like to thank Dr. Amy Schmid and Riley Hackley, my advisors, as well as my honors thesis faculty reader, Dr. Tom Mitchell-Olds, for their invaluable feedback and support. I'd also like to thank Dr. Lauren Luger and Joey Prinz, and thank you for listening to my talk. If you have any questions, feel free to email me. My email is below, avery.fulford@duke.edu. Thank you very much.